So quick introduction to remind people, I think everybody knows you, but in case new people watching might not, and then dive in. Yeah.
Sure. Well, I'm DeAnna.
I'm Kelly.
This is my husband that you don't get to see very much. And we're here just basically to talk about how we can harness the power of nature using indigenous microorganisms, or IMO, from your farm. So that's what we're here to talk about today. So thank you so much for inviting us to share with the community. We're pretty excited.
Yeah, I'm very excited. I know a lot of farmers from all over have been asking about this and excited about this because they are, as some of them said to you, they just know this is the answer because it's from your farm, it's native, it's biological, it's all of those good things. So
Yeah, the power of nature. Nature's balanced. Nature's perfect. It's got it figured out already for us. So
So that's what we lean on. So just to kind of give you a little bit of background of some context, right, of where we are at. The star here south of Minot, that's pretty much where our farm is, right there. So come visit us, Shorty. Now you know where we live. So that's kind of where we're at. A little bit about our environment. We are dealing with about an average of 14 inches of annual rain. Last year we had 4 1-inch rain events. The year before we didn't have any. And the fall before that we had 27 inches in September.
during harvest.
So it's it varies a lot. But for our area, the annual rainfall should be 14 inches. Annual snowfall should be about 40 inches. We are insulated still under about just probably under 3 feet of snow currently. So we got a pretty good blanket on us yet. And we have 134 frost-free days. We grow small grains in our rotation: winter wheat, spring wheat, mustard, flax, oats, some heritage wheat varieties, spelt, and also heritage barley. We like to use some companion planting in our system when we can. We like an oats, mustard, and clover blend a lot. And we like to put clovers in with our small grains in between the rows as a companion plant. We like cover crops when the season allows it. And also in our system there, we don't use any seed treatments, no fertilizer, no manure, no compost, no insecticide, no fungicide, and no pre-harvest desiccant. So that's a little bit about what's that?
That picture there, I guess, just a little bit about the picture. That's a Rouge de Bordeaux facultative wheat. So that's 1800s wheat genetics we're growing at scale. I guess just to give you a little context of what the crop is there in the picture.
it's beautiful. It really is a beautiful picture.
It grows beautifully, and the root mass has so many benefits for us on the soil side. So we do like it, and more of our acres. I don't know if I mentioned, but we farm 2,000 acres in about a 2-mile radius. And so more and more of our acres are being shifted to heritage varieties. So I'll let you tackle this one.
Well, this was kind of a turning point. So we reduced to eliminate fertilizers on our farm over the course of 5 years using basically Dr. Christine Jones method. So over the course of 5 years, we reduced down to net zero. And we saw things really improve. So then we started searching out the why. So we saw the results in our field before we started to really understand why this was all functioning. And Dr. James White from Rutgers University has done a tremendous job explaining this rhizophagy cycle on how our plants are actually cycling microbes in our soil system. So they're not only consuming whole bacterial cells and stripping their cell walls off and using that as nutrients, but they are then putting them back out into our environment to rebuild themselves, where they will continue to divide and multiply in our environment, only to go through the plant and do it all over again. So it's a completely regenerating, recirculating system. So this really brought to light like how this was really happening. And we discovered this when we had already eliminated our fertilizer in our farm. So one other key metric here is this process, and most biological processes will be stopped by spoon feeding nutrients to our crops. So we were kind of at a really beneficial, critical point in our transition where we had already eliminated our fertilizer when we jumped on with this, or understanding this method. So we had everything set in place to already engage in this process rapidly. Now, and this process under high fertility wasn't allowed to happen. But now we're able to expand that, repopulate that, and we're simply cycling cells through our soil system without applying anything. We're not applying micros, macros, anything anymore. It's all gone. I think that basically explains. I feel, is everyone familiar with this process? I feel Dr. James White's on the forefront of this and doing a fantastic job explaining.
How long? Sorry, Kelly. How long did it take before you went to 0? 5
years.
Okay.
Yep, we reduced over the course of 5 years.
Mm-hmm.
But the initial year one, we had 0 fertility on approximately 500 acres. During that transition time, we had it got really complex. We did variable rate fertility, variable rate soil testing, variable tissue testing based on soil type, soil sampling based on soil. We had stacks of tests, stacks of data to look at. And when it got right down to it, it was just plain pure reduce. Just take an easy button and just reduce. You can take one test and reduce over the course of 5 years, because the data really wasn't telling us anything. The data was leading us in the wrong direction. The crops were leading us in the right direction. So what we had observed on 2 years' time on that crop with no fertility was we had less disease pressure and we had less insect pressure, which less disease and insect pressure meant less work for me. If I got rid of that off my farm, things could get easy. And that's what I was looking for, was things to get real easy for me. And it did. It got real easy when there's nothing wrong out there anymore.
So you kind of had to take a leap of faith, right? And the data is telling you, and I'm sure all the experts are telling you, oh no, you need all this, and you, because of the science, and you knew what could happen, it was an informed leap of faith. It wasn't, but that's kind of what you had to do, right?
Yes. Yeah. And the experts, that's a good point there, because it's what experts do you lean on?
Yeah.
Dr. James White is an expert in this, and we shifted our mindset to he wasn't selling us anything here. He was providing us with the information.
Well, and we now had the science to back up what we were observing in the field, right? So we had observed that our plants were growing without spoon feeding nutrients, right? We had observed that, but we didn't exactly know why or how it was working. So when we came across this type of science, it was just like, okay, so now we have some proof, like some explanation to go with what we're observing in the field, because we had seen it in the field, we just didn't know the science behind it. So this was really important for us.
And the yield really didn't dip down that much where there's no fertility, because the disease pressure was less, and our rains were becoming limited. Our environment was being stressed, so those areas handled the stress better as well. So during that time period, we didn't go through ideal growing conditions, so to speak. So we didn't really see a yield hit where we had reduced to net zero. So then we just rapidly scaled that up on the rest of it. We initially went after the high organic matter, so anything 3.5% organic matter or higher.
And we were growing crops that needed less, that weren't intense, like mustard. It's kind of a low input crop as it is, yellow peas, pretty low input. We kind of shifted some of our acres. We don't raise any corn. We don't raise any soybeans either anymore.
And we stopped soil testing and we stopped tissue testing.
Right.
And because we had healthy plants, you know,
Mhm.
if you're healthy, you don't really go to the doctor. You know,
Just as a recap, we had a few people join. So you did this test on reducing or going to zero on 500 acres of your 2,000 acre farm for 2 years before you expanded to the rest.
No, it was just 500 of the acres based on organic matter were cut cold turkey off of fertilizers
Okay.
as a whole. And then the other 1,500 acres were reduced to eliminate 20% a year
over the course
over
of
the
5
next
years.
5 years.
Great. Good.
I was 3.5% organic matter or higher. I had felt that I could grow a reasonable yellow mustard crop, a flax crop, a pea crop, a wheat crop on that ground based on solely organic matter. You know, we didn't really understand any of this rhizophagy process or anything like that at the time. We just went by the organic matter number because our soil test data and our tissue test data wasn't lining up.
And we were introducing cover crops at that time,
Yes.
where it was available, right? So when the season allows, we like to put a cover crop in. So we were also leaning on that diversity from.
We were pretty successful following field peas with cover crops. We had timely rains happening after harvest, so things really worked well for cover crops for quite a few years there. You know, until the rain shuts off, then it's a whole different ballgame. But the rains were coming, cover crops were working really good following field peas. So we had a nice diverse 6-way mixes going in the fall, constant cover. You know, things were working good.
So what is IMOS? Okay. It is an indigenous microorganism solution. And up until a few months ago, it really didn't have a name. So we were encouraged by some of our mentors. Well, one in particular, he's a little pushy guy from South Dakota. He had suggested we name it. And so we did. So we had been leaning on biology since 2019. So this is we're going into our fifth season of applying what is now called IMOS, biopriming our seed. And there was right around 2019 when Kelly pieced all this together. We were wanting to do
It's more the winter of 18-19.
Sure.
Yeah.
Well, we had been following Dr. Elaine Ingham and the compost methods.
Biologically complete compost
Yep.
method.
And the Johnson Sioux bioreactor. And so
Natural Korean farming.
Korean natural farming. So we had been kind of watching and learning from all of those different examples. And what it came down to with any of those was that it was too complicated for me to handle by myself. So at the time, our kids were then 4 and 2. So pretty busy around home for me. And for me to be able to commit to learning how to keep a compost pile where it needed to be, the labor, I didn't have the labor. I didn't know enough to make sure there wasn't any huge implications with that. And also it was just too huge of a commitment. And the Johnson Sioux bioreactor, we love the idea of that. It works, like people are using it, it works. And I know Shorty has a lot of experience with it. Again, it was one of those things where it was a year out. I couldn't commit to keeping it warm. I didn't know at the time how that worked with freezing temperatures and things like that. And then watering it was just a huge commitment and too many days in a row of having to take care of something when I was already trying to keep 2 tiny humans alive. And I just didn't know enough.
And I couldn't wait a year. Like no.
And of course. And so I told Kelly, I was just like, if we're going to do this, it needs to be something I can manage on my own. And it's not that I'm incompetent. I just needed it to be simple enough that I could also handle it with everything else that we had going on. And so I just told him, I was like, if you want to do the biological aspect and be putting it on seed, you need to make it simple. And whatever you come up with, it has to be something I can do by myself. He runs the air seeder. And so I knew that meant that it would be me creating the solution and applying it, biopriming it on the seed. So it had to be something I could manage by myself. And so he developed a recipe of indigenous microorganisms from our thriving system, and a system of brewing it for 24 hours. And I apply it to seed. It's as simple as it sounds. And it brews for 24 hours. I can do anything for one day. And so that made it easy for me to make this happen. And I think that we know it works. And it took a lot of me convincing Kelly to start talking about it with other farmers. And it took a lot. And the first farmer we started talking to about this was that pushy farmer from South Dakota. And he was just like, this works, it has to work. And I was like, well, you haven't tried it. He said, I don't need to try it to know that it works. This system is what he had been waiting for. And so he tried it on his farm last year, and encouraged us to continue talking about it. And there's a little bit of, it's kind of scary to be talking about something that nobody else is talking about. And Kelly's already kind of a rogue farmer, and kind of on the outskirts and doing things that nobody else is doing, but talking about it is something different. And the idea of bringing it to other farmers is important to me, because I want other farmers to be able to experience the freedom that we have from eliminating inputs or reducing inputs and leaning more on indigenous biology to make that happen and to produce our crop in a way that is manageable for us as a family. And so I pushed and pushed and pushed and pushed. And you don't know what that's like.
Oh yeah. Oh,
No.
yeah, I know.
So and we developed a video series. It's a simple
And-
5-part video series to help farmers harness the power of nature.
And all of these other methods do work. So, I mean, if you can pull the Johnson Sioux off, I mean, it works. If you can do the Dr. Elaine Ingham method, it works. I mean, any of these biological methods are going to be a substantial improvement over the agronomy center, hands down, without a doubt. And I guess we weren't talking about that here. I mean, I was a little selfish. I guess I wanted to make sure, if you see up there, like there's fertilizer infrastructure on our farm. So I had to make sure that was all sold and gone off farm before because otherwise there's no one out there to buy that stuff. So I did sell all the fertilizer infrastructure, the storage, the equipment, all that before we actually started talking about it, because that stuff's going to be worthless if nobody's using it anymore.
Well, also, I think one of the things I wanted to make sure I understand, I think this is true too, but you're not just reducing a dependence on the synthetic, you're also not having to switch from one input set to another. You're not having to buy other biologicals. You're creating your own, right? Because there's a whole new bio industry that's like giving you bugs in a jug and, you know, all of that.
No, that's a really good point, right? So all of the industry is saying, okay, you're spending X amount on your fertilizer program. They say, use that and take some of that and apply it to biology, right? So take some of your allotted fertilizer dollars and put it towards biology. There's not one part of the industry saying, hey, guess what? Maybe you could use a part of that fertilizer dollar and put the rest in your pocket. No, nobody is saying.
Oh, they're not going to fund their own irrelevance. They're not going to do that.
Nobody's saying, hey, you can actually reduce what you're spending.
Yeah.
They just want to keep farmers Farmers are
Yeah.
used to spending money and they want to keep them spending money and just switch the way there's,
Who they're spending it on. Yeah.
and it all goes to the same companies, correct? So we won't get in too far into that. So that's really what it's about, was giving farmers the opportunity to be able to do this for themselves, right? That's all it is. It's putting the information out there so farmers have a choice to what type of biology they're leaning on, right? Which is why the Johnson Sioux bioreactor is so great, which is why Elaine Ingham's compost method is so great. It's teaching farmers to do it themselves. And this is another option for that.
Because nobody's making new microbes. That's, I think, some of these companies are saying that they're bringing in something that's new, and nobody's really making new microbes. They're all being sourced from our environment. And I feel our method empowers that farmer to go to the holy grail. I mean, healthy soil will provide us with everything we need. Absolutely everything. Our micros, our macros, a well-structured, well-covered soil is everything. So that's what got us to go right to the holy grail, right to the source, everything right to nature that had it already figured out. I mean, it's such a complex system. You can sit there for hours and hours under the microscope looking at that, trying to understand it. You'll drive yourself batty.
And it's important to know that it's living, right? Everyone, you need to see and everyone should have the experience of being, in the microscope and seeing what's happening. We don't have to exactly be able to point out what's happening. If we're borrowing IMO from a thriving system, I'm not too worried about what types of populations are there. Just about a balance is really what we're shooting for. So we developed the video system. Like I said, it's a 5-part series. It takes about 3 to 4 hours to watch in its entirety. The first one is just an introduction to why, like I talked about just a few minutes ago, why we developed the video series and how it can work on your farm. Video 2 talks about how to borrow the IMO and learn where to source it from, how to borrow it, how to feed it, and how to return it. That's really important about IMOS, is that we aren't extracting anything from our native system. We are borrowing it, repopulating it, and then returning it. And that becomes really important to finish the cycle. Video 3, Kelly breaks it down on how to build a brewer. And again, it had to be a step-by-step instructions. So if I had to watch the video by myself, I could go into the shop with the tools and make it myself. And so it's very detailed. I have a typo there. Very detailed, very easy, and so simple, even I can do it. Video 4 is just how we brew IMO. So IMOS is, and we feed it, we reproduce it in a 24-hour period. And then video 5 is how we apply it. And the best ways that we've learned on how to bioprime the seed. So it's a simple 3 to 4 hour video series that you can watch and rewatch again, and what's important, why we're doing, I don't think we always will have to do this, right? But, and that's the idea. That's also, our system is very inexpensive, because once we introduce the biology and keep a home for it, I don't think we will always have to do it. But as I explained before, we are insulated here under 2 to 3 feet of snow. My microorganisms in North Dakota come out of dormancy, come into spring looking like this guy over here, grumpy, tired, rough,
Tough,
just a little bit gritty, right?
Ready to make your day.
Right. And then we have the picture over here, like the microbes in Ohio. They wake up being fully fed after all winter. They're ready to party. Everything's sunshine and rainbows. They get rainfall by the inch.
We measure
So like I said, also
in Ohio, they measure their cover crops by the tons of biomass they produce. We measure our cover crops by the successful of our germination rate. So
it's a completely different microbial community. But the point is, it all benefits from added biology, right? Of
Yes.
indigenous microorganisms, right? So while we might not have these, microbes in our soil that are like ready to party and ready to produce for us and like all happy and sunshine and rainbows, we can wake them up and get them to participate for the season.
And those easy ones from those other climates might not make it here. They might not be able to survive our winter. They might not be able to survive further north. So we take something that is proven, that's working, that has survived our climate.
And so that's why the seed becomes a super important carrier of the biology. So we can start our seeds off right with IMOS.
Deanna, sorry, but UC had a question in the chat a little bit ago, asking about, so the testing is understandable that you kind of had to disregard the chemistry of the testing early on, but have you done testing after the fact to kind of see what's there, or would that be beneficial to see if the plant actually has adequate nutrition, or does it give kind of totally skewed results even after the fact? And secondly, he said that he's using this Solvita's soil health suite test, and he's just wondering if these Regen IMOS results would show up in that test.
I have, since we went to net zero, I have used, now I don't want to downplay any of the soil health tests. They're all a tool. If you're used to looking for a test, they are a tool to get you to reduced. None of those tests are 100% accurate. These populations can change within 20 minutes. Moisture comes, these populations can explode. I mean, you can be looking at something 1 trillion times different within a 24-hour period. We did do one of these soil health tests, and I'll tell you right now, the soil health told me to put down fertilizer. So I did. I went and purchased some fertilizer, and I put it down on I split a 300-acre field into right down the middle, put fertilizer, phosphate, went back to phosphorus, nitrogen, sulfur, zinc, and I think there was one area of potassium. So we applied the fertilizer like the tests said. We went into a stressed growing season, moisture stress, heat stress, and we just watched those plants. We did not do tissue testing, but we watched those plants. Root structures on the fertilizer side were small. We were back to a bacterial infection, because we were dealing with bacterial infections in our cereal grains that there was no cure for. So the bacterial leaf streak was back in the wheat. We then had aphid pressure. We had hopper pressure. We had plants that were shorter. And as the season progressed, our side with no fertilizer actually was darker, greener, and taller. And then when we went to harvest that field, the side with the fertilizer turned red on the yield maps. So we actually yielded less where that fertilizer applied. So now here I paid for all the testing, I paid for the fertilizer only to yield less and all the extra inputs. So that soil test did work for me, because I went home after harvesting that field and I sold all of my fertilizer infrastructure. Not only did I sell the fertilizer infrastructure, I sold the fertilizer that was in the tanks. I still had the liquid nitrogen there just in case this might not work, just in case I might have to go in and top dress something if I saw some nutrient deficiencies. But after seeing that, it gave me the confidence to go and sell the infrastructure off the farm. And I didn't even apply the nitrogen. I sold the nitrogen to a neighbor.
Yeah.
But that's great, because it is science. You're able to really prove, have a hypothesis and test it and see the difference. And so you're making a fact-based decision. It's just not the kind of infrastructure and testing that
we're used to, really.
Yeah.
Right?
Mm-hmm.
So it basically gave me the confidence.
Yeah.
And I think that's what those tests are designed to do, is give you the confidence to pull back the reins and let nature take control. I don't think we're going to be able to measure this and quantify this. It's a living, breathing, changing system. So I don't think we're going to get to the point where we can completely identify everything that's out there, how it functions from a test when it's ever changing. But I think to give you the confidence, that's where those tests really shine.
Right, as a tool.
As a tool.
And in I think that it just kind of all depends on how we apply the information and where that data goes, right? So
And right now, I'm not looking at a test, but I'm looking at my wheat plants and they're healthy and they're green. Our yields are good. Our quality's good.
and
I'm kind of afraid to look at a test because it might tell me to go out there and fix something.
and we test outcomes, right? So we're working with, Jill and sending our grain off to her at the end of the season to look at the outcome of the grain based on our growing practices, right? So we are looking at outcomes. It's just we're looking at the final outcome, which is what we feed to people, what we feed to animals, and how we've, how the plants have fed our soil throughout the year. So.
Okay, I need to ask here now, Jussi is here. Actually, if we get the biology working better, would it actually be the case that the efficacy of those nutrients plant is getting via bacteria or whatever systems, the efficacy actually may go up? So that if we are only testing the sap analyze, we are just checking the raw material getting into the veins of plant and not necessarily to the cell level. So could there be something like this benefit also, which is not really able to be seen that easily as a conventional guys would like to see it?
Yeah, I think that's really accurate. Like, I think that we are so far away from knowing for sure what's happening. And we are not experts on microbiology by any means. But what we are is experts in our own field.
And I think everyone needs to be experts in their own field.
Yes.
I don't think anyone knows that farm better than the farmer that's on that ground.
Yeah. And we all need to be making the decisions for our farm based on what information is available to us. So and there's more and more coming out all the time, which is really, it's a really exciting time to be in farming and to realize that we can impact. I mean, farmers aren't just growing food. We can heal a lot of the problems that are going on in our farming practices and influence that in a better way. And I think that biology is one of the ways that we can do that for sure.
And I think we can almost get to the point of knowing too much, like nature's something that can't be taught, but we can learn from that. So we don't get into that prescriptive biology and kind of follow the same metrics we've done with traditional agronomy and try to control it. But I mean, it's fun and exciting now to understand, through some of these scientific studies and everything, how this is all functioning, where it's coming from, if that's getting into the cell and that sort of thing. But I feel I got to use a little bit of caution, so I don't try to find out one silver bullet and try to hone in on that and go down the prescriptive biology path.
It's almost like you have to hold some of the mystery and the awe of nature along with, yes, there are tests to be done. And yes, there are, particularly outcomes looking at that. But, you have to just hold some space for the fact that this is all a miraculous thing that happens, all this life that's happening and that you're stewarding. And it's part of that. I know you guys talk about a lot, the shift in the mindset to regenerative and seeing yourself as like this, this steward and supporter of nature rather than the guy in charge of everything. It all depends on me, and so I think maybe it seems like that's some of what you're getting at, like that there's a piece of it that you're never going to fully dissect down to all of its parts, and that's okay. That's the way it is.
Right. And it's humbling to realize that we don't know it all.
Yeah.
We don't need to know it all. And some people are okay with that and some aren't.
Yeah.
And so they're willing to get the information from wherever they have to get it. So our goal, Kelly's goal is to master the art of doing nothing. I make that difficult with Guardian Grains and our food company.
Yeah,
But-
you've been failing on that one. I'm
Yeah.
sorry. I
And we got all this extra time because we're not caring for the sick crop. Now we can start doing.
Well,
Now we
that's
can
true.
start
That's a
caring for
great
the
point.
people, right?
Mhm.
And so it's just an evolution of the farming system, right? And trying to do the next thing as we're called to do it, right? Because it's a huge leap of faith to be able to trust the system. And we can do it because we trust, like for a long time, we trusted the agronomy center. We trusted our seed salesmen. We trusted all those. And we paid them to trust them. Trusting nature is something we can do for free. And it's something that we can do on our own accord and be further connected with our soils and what that means for us and growing better food for people and animals, right? And so it's a pretty awesome thing to be in this position for sure.
So you are using a farm saved seed, I guess.
Yes.
So have you noticed that the seed actually adapts somehow better to your own soils over the years? I mean, if you would compare the same pedigree variety grown in, let's say, that Iowa or back home in northern Dakota, would that actually give you a different result if you were to look at the bacteria content which is around the seed? I mean, the adaptation to the local conditions. That do you feel that would be one of the important things?
Yeah.
Yes,
Yes.
yes.
We fully believe in seed transferred
Endophytes.
endophytes.
A lot of those are contained right within the seed.
Yeah.
And we have brought on just a trial. We've brought on foundation seed, which is first generation new breeding seed propagate on the farm. So we've tried it. And I will tell you that bringing in foundation seed, something from off the farm, will not handle the natural environment like the seed that's being saved and grown and propagated on the farm year after year. We entered last season, we had 4 1-inch rain events, and all of our crops did really well. We had a really good crop last year on 4 1-inch rain events. But the foundation seeds that we did have on some small acreages did not grow near the height. I mean, we were running stripper heads on the ground, just getting that foundation seed, because I mean they were short, but those varieties have all been bred to be shortened up because of the high nitrogen load being put on them. But they did not handle the 4-inch rain event, 4 1-inch rain events during the growing season like the stuff that we saved.
Yeah, and it definitely plays a role. And so anytime we introduce a new variety, and now when I say, like he mentioned, we had some barley and oat varieties that are foundation seed. But even when we introduce new heritage varieties, so like we brought in a turkey red winter wheat, and planted it on the same day as we planted our Rouge de Bordeaux in the winter, which the Rouge de Bordeaux was saved from our farm. We put it in the same day as we planted the turkey red that we got from a seed salesman. That was grown in an organic system. We put it in the same day. The Rouge de Bordeaux in our system that had been saved seed was emerged in 7 days. And like I said before, we farm in just a 2-mile radius, so everything's very close. But this field from the Rouge to the turkey red was just directly across the gravel road. And I think it was nearly 50 days, 50 for emergence on that turkey red winter wheat. And it was extremely dry, right? It was extremely dry that fall when we put it in. Our Rouge de Bordeaux was accustomed to that and used to our system. The turkey red winter wheat was not. And so we like to save seed as much as we can. When we're introducing new, we do notice a difference.
That turkey red came from an organic system, high tillage, high animal manure input system as well. So I think this is going to be a really important question to ask your cover crop grower. So your cover crop grower, the source you're buying your cover crop seeds from, if they are doing more of a biological approach, growing those cover crops are going to come to your farm with this microbiome already established. So I think we can do a lot of transferring of endophytes just within the seeds coming from those farms. So, I mean, if those farms are engaging in this process, those plants are going to give that communication network or establish that biology on your farm easier than something that's coming from a high yield, high synthetic output system.
This is fascinating because I know with Craig Cameron, who we have on, who's a rancher in Canada, and Jill works on a ranch in Kansas, and they've talked about the genetics of the cattle being suited to the land and the system, and that that it's so much better when they keep some of their native herd as opposed to bringing in a new, even the same variety, same genetics, but not the same. And then those cows do worse because when they were being formed, when they were, they were being fed the nutrients from that system. And it's the same with the seeds. I mean, it's like the seeds are being created in the system, and what is creating them is this there's such a difference in the biological approach that it affects the seed and what it's amazing.
Like Kelly mentions the cover crop seed.
Because we're going to have to bring some seeds onto these farms, because some farms are in an area where they can only grow a certain amount of things. That's what the markets.
That's what the markets dictate.
So they're going to have to bring some of these cover crop seeds onto their farm. But I think it's going to be very important for them to ask these questions on how those seeds were grown.
Right. And cover crop seed, because we're putting it in usually a pretty diverse mix of plant species, becomes a great carrier for biology and for IMOS, because we can put it, usually cover crop seed is untreated. This is where people run into problem with putting biology on the seed, is if they're using treated corn or soy or sunflower seed. And cover crop seeds can be purchased that naked, right? And so you can apply biology and get it out there in
on a diverse mix
huge
of plants
numbers.
that support this whole community.
Yes,
in
and
huge numbers.
and that'll bring us on to quorum sensing. Kelly likes
And
to
also
talk about quorum sensing.
with the fall cover crop, I mean, that's the best time of the year to establish biology. And when those fall cover crops are going on, the farmer can then apply those with no fertility. So they can put no spoon-fed nutrients on those fall cover crops, to lessen the economic impact on their cash crops. But that would be the way to get started in this.
And it won't interrupt the communication between the plant and the soil microbiome with spoon-fed nutrients. So we're giving it open communication between the plant and the soil. So
Epigenetics is the word Craig Cameron helpfully saved me in the chat that I was struggling for. So that's
epigenetics. Yes,
what you're doing. Yeah, affecting.
Mm-hmm.
Okay, so quorum.
So the quorum. So this is sheer numbers. So the intercellular communication is dependent on high density populations. So we have to get these bacterial numbers, these fungal numbers up high enough to induce them to communicate. So when we get them so concentrated that those cells are rubbing each other, touching each other, bumping into each other, then they're going to initiate this communication network and be able to talk to each other interspecies. So you'll have different species be able to communicate with each other to perform different tasks. Where if you have all these different species at low concentrations in your soil or around your seed, they will not perform the same as they will in group behavior. So when you have group behavior high concentrated, they'll be able to perform things that they can't do as individuals. You can kind of think of like bioluminescence in a shrimp. It's a bacteria that gets in that shrimp. That's not a shrimp. I messed that up.
There's a squid.
It's a squid.
Shrimp,
squid.
what,
But
anyway,
when they get confined in that little area, they're able to produce light. So which is something they can't do on their own. So the same thing here, we get a diverse group of microbes, highly concentrated. They can perform and do tasks that they can't do under low concentration. This aids in a symbiotic process to provide exponential cell growth, division, and this aids in nitrogen fixation. Because now you have different species working, communicating with each other, telling each species what the other needs, what it wants to build, and then they have someone else come in telling, well, this is how we can do that, and they work together to build whatever structure you need. And that's what led us to IMOS, the active aerobic brewing, is because that is intensely high concentrated. Like if you look at that, those cells are stacked on top of each other. They're bumping into each other. So we are putting an extremely high density population of native microbes on that seed to initiate that process of quorum sensing. So initially, right from germination, we can engage in rhizophagy process. So those cells will start circulating through that plant and growing out into our soil system as those plants grow under high population. Okay, here's kind of a little, I thought this was kind of fun, a little cartoon graphic of how we can bring a quorum of indigenous microorganisms to the seed surface. So this is kind of a little cartoon signifying what they can do. So the bottom left picture, every one of those microbes has a light that needs energy. So when they're wandering around by themselves out in the environment, they might be there, they might show up on a test, they might show up on a DNA test, but that they produce energy and they're out there in our soil, but there's not enough of them. We get them under high density, high population environment, then they can work together and produce the energy. So even if they're there, if their numbers are too low, they won't be able to perform the task as they will under concentration. And that top cartoon there, I like that because even like you look back in the corner, just to reach the light switch to turn it on takes 3 of them. So if there was one there, he couldn't reach the switch to turn it on, but 3 of them together can reach the switch to turn it on. But now just to install the light bulb, there needs to be 3 of them just to reach the light bulb to turn it in. So we got light from the energy that's the switch is being turned on.
and this impacts everything. This impacts immune system function in our plants. This impacts nitrogen fixation. Like this can impact the plant on every level of life. If we have a diverse group in large numbers working together to influence our plant's ability to produce on its own at the seed surface. Do you have any more on that one that you want to talk about?
No, that's all under high concentration. So this is why it's valuable. So initially during germination, we come in with very, very high concentrations. So when that seed's born, when it germinates right away, it's got that entire group behavior. So that group can go in and colonize that plant initially from the get-go. So the first roots that go out into the environment, if we are engaged in rhizophagy cycle, those cells will be going inside that seed, out those roots back into our environment. So even though we applied a high concentration of biology on a seed this big, as that plant grows and is dependent on that microbial community, those root structures will grow bigger. So now all of a sudden we get a root mass, say this big in the soil. Now that high concentration of biology is now covering an area this big in your soil because they have now been repopulated and put out with that plant. So even though we applied a small little area, that affected the pH where that plant was growing, affected everything where that seed was growing on those roots. And our yield is determined approximately 14 days after germination. So if we're going to biologically feed these crops, we have to ensure the delivery of the biological nutrition initially at germination if we're going to supply that during the entire growing season. Now, if we were going to have a starter fertilizer or something here to get this plant up and going, we would shut this process off. and they aren't going to be engaged from initial germination to support that plant through the remainder of the growing season.
And the rhizal sheath that you see on these two pictures, this is the proof that you have your biology working here, creating glues.
These are our tests.
Yeah.
I guess this will be tests we perform.
Yep, creating glues, working together, building soil structure, right? This is proof that there's air in the soil. We have to have air space for these microbes to breathe, for our plants to breathe, for the soil to breathe, and we need air, water for water delivery. So it becomes really important to get as much root mass out there as we can to create those gaps and spaces for all of these underground communities to do their work, right? So I've heard them referred to as essential workers, and they are, right? So because we completely rely on our underground community and the underground livestock to supply our plants with nutrition, with immune system function for the entire season. There is not one Band-Aid that we apply, and not one way we're going to go in and try to fix anything. So by applying the biology to the seed, that gives us the best chance to make sure that our microbes, like you saw in an earlier slide, that look more like, you know, rough and tough. They don't all look like Brandt's microbes in Ohio. We need to reactivate them and wake them up. And this is a good way to do that. And it really helps create a disease suppressive system for our plants.
And a bacterial biofilm has a pH around 7. So I mean, we can actually buffer that zone right where that plant's growing. I mean, you even look at a young little seedling in the middle. I mean, we've already established a pretty large area just off of a little bit on that seed. You know, we've now expanded into a larger area. So it expands rapidly. Like, it's not something you're going to have to continue to do. You do have to, however, continue to provide them home and shelter and food. So they're going to need plants to keep them fed. They're going to need cover on the soil. They're going to need soil structure. They're going to need those glues to form soil aggregates, those big clumps and rocky structures in the soil so that water can infiltrate and oxygen can get in, because these guys need to breathe. They're not going to survive in a compacted waterlogged soil. You know, we've got to build them the home. And if you are coming out of that, the compaction and everything during a transition, it's going to be more important than ever to continue to apply until their homes are rebuilt.
And that concludes our little slideshow about IMOS. And so now if we have time, if you want to ask questions or whatever, these are just some ways we can get connected. I'm on social media. Kelly is not. And our website is there on the bottom, and there's a QR code to bring you to IMOS there too. So we're happy to answer any questions. Oh,
I guess last month we decided to make this even easier. We put together these little do-it-yourself kits, so farmers can now take these and produce their own biology on farm. So everything's been pre-measured, pre-done. This little package here will do approximately 1,000 acres. Okay, so when we're looking at the amount of material you're handling and everything, this will do 1,000 acres of you sourcing your own soil from your farm.
That's amazing. That's
So
just
this
amazing.
is seed treatment. So I'm talking wheat seedling, you know, biopriming wheat seed, but that'll do approximately 1,000 acres of using your own biology. And that's going to get you out on your own farm, digging in your own soil in those native areas. So you know what that soil is supposed to look like. You can see that soil is undercover. You can see that soil is aggregated. And once we're there, then we can supply everything that plant needs. But you're going to look for those spots on your farm. So we've made this as easy as possible because everything's pre-done, pre-calibrated for a 30-gallon brewer that you can build with hardware store supplies at any local hardware store anywhere around the world at a very low cost.
and that was also suggested to us by a pushy guy from South Dakota. So he wanted us to make a kit for him, and so we did. and I made it even that much easier. Everything's in a water soluble pack, pre-measured for a 30-gallon brew, which does roughly 600 bushel of wheat seed for me. So it does about a truckload in about 30 gallons applied to the seed. And everything's very easy, and it's in a biodegradable bag, so you can just throw the bag right into your garden or in your compost pile and not have to worry about it. So it's as little waste as possible.
Yeah, you don't even have to open the packages. They're water
Nope.
soluble packs. You just drop them in, they dissolve. I mean,
No measuring, no nothing.
because that too many times we've left conferences and everything, and everyone has a lot of ideas, but then it gets to be too much work.
And
So-
guess what? It's going to be easier for me this season now that they're all pre-measured and pre-packaged. It's going to be super easy for me because there's going to be no measuring involved on my part, and it's going to be just make it that much smoother. So I was just like, way to go. You could have done this for me 4 years ago. It would have been nice.
Yeah, but continuing to improve and make things easier is what we're.
That's right.
What we're saying. And once you start working with biology on your farm, I mean, things get really easy, real easy.
Yeah,
Things
so
start
easy. You
clicking.
can start a food company.
Yeah, you don't have to. You will have
Well,
the-
yeah,
Go ahead.
let's leave a little, we're almost at time, but we're going to keep going for any of you that want to hang around, to ask questions. I'm sure there might be some, and we have a couple of new members, which I'm really thrilled to have, Mark and Avery. So if you guys want to ask a question, you might be on mute. So just click unmute and ask away, or you can type it in the chat and I will.
Yeah, and
Or-
if we can have an open conversation, that works really good. Because that's how we learn best, is through others. So if
We're
we can-
still learning. We don't know it all. We know
you know nothing.
a fraction of what's going on down there.
Right.
That's the fun part.
Right.
So questions?
I would
Well,
like
this
to
is
ask
very,
something.
very nice
Sorry.
to listen to this. I can tell you that I got to know a professor lady nearby university, the microbiological professor, and she happened to be a farm wife as well. They have cows at home, and lady works at university. Now we are in the project to. She has a few doctorate guys to have a doctorate study, and I agreed to find out them this kind of comparisons where we would have a kind of a good place and bad place, and they are going to take those samples down to their own laboratory environment, and then they are doing the RNA sequencing and all that stuff they can. And hopefully next winter we can talk about that, the first results here as well. So yeah, it's kind of interesting, really.
Yeah.
I'm really happy to be involved this way. Yeah.
I also want to just put in a plug, shameless plug, for the Grounded Growth Paddock, because DeAnna and Craig, who's on, have been star students over there. And what we're trying to do is give farmers the ability to do some marketing for themselves. And so, DeAnna put together this video series, and she's got her great website, and that is not all at all related to what we're doing, but we're learning together. And I had to learn about marketing when I launched my own little company. And so I'm helping similar kind of concept, like helping you to not need marketing over time, and not to hire it out. Although it's very nice if you can. I would recommend it if you can, but I think it's a wonderful example of what DeAnna has done, because you have this knowledge, you have this great knowledge, and it's worth a lot, but packaging it, putting it into the video series, putting it on your website, having a cart, having all of that is important. And I think more farmers should do that. And I think they're sometimes held back because it's intimidating. It's a whole other world. So over in the Grounded Growth Paddock within our community, you can watch the videos about DIY, learning how to build your website, build your social media, build your sales funnel, all of that. And you don't have to just sell your products online. You can sell information like DeAnna is doing. So if, especially for the new folks, you might want to check that out, and encourage you all to join us there whenever you have marketing questions or messaging questions. So any other questions before we wrap up? And it's a lot for people to digest, and as with all of our presentations, we are recording it and we will put it in our library so you can watch it again. The regenflix.com. Eventually that's going to become a thing. I know. Like
Yeah,
Netflix.
it's totally a thing.
Regenflix. It'll take a while, but it's
Is
okay.
there anything we missed in the chat, Sarah?
You did have a nice comment from Mark Kopecki, one of the new members. Terrific information. Thank you very much for sharing your experience and your understanding. A lot of great comments from Shorty talking about how wise you guys are and especially the connecting to nature, piece. I think, there's a comment from Anne, and she asked, how has weed pressure evolved? Do you rotate crops, and what do you do to volunteers in an organic no-till system? What are your yield levels? So that's a great question.
Well, we're not, we're no-till, but not organic. So, that being said, with the removal of the spoon-fed nutrients, with the removal of that, the weed pressure has gone substantially down.
And organic nutrients too, because high levels of animal manure put on will stimulate
Weed growth.
weed growth.
So we don't have that extra pressure is not there. And also the challenge for us is to be able to get into our seeding system before the weeds come, right? Because if we can get in there and get the crop canopied prior to the weeds coming, we are in pretty good shape.
Yeah.
But I will tell you though, just looking outside and looking at 3 feet of snow, it's going to be very difficult to be ahead of the volunteer plants that are coming this spring when we finally get out there to seed. So there may be a herbicide pass. We are not organic. We use very little herbicide and only it's on a field by field basis. So nothing gets the same treatment. It just really depends on what's growing there, if it's going to inhibit our moisture, because like I said, we are running into water being our limiting factor. So, and while we can have companion plants out there that are helping our cash crop, if they're also taking water when we only have 4 1-inch rain events, it can really be detrimental to our when we're during harvest, and that'll show up on the yield monitor, right? So if we go in with a herbicide pass, it's early in the season. But like I said, a lot of times we don't need to, because we have companion plants in there with clover or a group of plants growing together. And so that eliminates the herbicide path.
And another thing, if our environment allows, 2 years ago in a D4 drought, none of our fall cover crops made it. Not one seed germinated. We were out of moisture. We were out of rain. So there we were left sitting going into winter with bare fields. Our biology was so active, we chewed through our residue through the winter under the snowpack. So come springtime, we had bare fields. And I was just ready to pull the drill out to start planting, as our soils were warming up and they were dry enough to get out there on the field. And we were hit with 5 feet of snow as I was pulling the drill out of the building.
Yeah.
Like we were one day, we're ready, everything, seeds in the truck, we're ready to
Yeah,
go.
I had brewed a batch of IMOS. I had bioprimed the seed, and the snow came and it didn't leave. 5
feet of snow took us about 4 weeks to melt off and to get back into the field. So during that time period, we can't traffic those fields when it's too wet, because the one thing we concentrated on was reducing compaction. Those field conditions have to be fit to be out there doing anything. So in that time period, with all of our residue gone, a failed cover crop, stuff's going to grow. Volunteers are going to grow, some weeds are going to grow. And we're going to have to, and we're in hills, highly erodible ground. So there's going to have to be some type of a method to clean those fields up to plant. We cannot roll or crimp that down in time here. We cannot till our land, because then we start all over building that structure.
Yeah.
But if we are going to use a herbicide, as a burn down, the main thing we want to ensure is that biological population is there functioning in the soil to degrade that herbicide. If those guys are missing, they're not going to be able to do that job to degrade that. So we have to make sure those biological populations are there. And I think in the event we're going to use a herbicide or something, that's where to be important to maybe reestablish some of these communities.
Right. And it's really been a benefit for us working with Sarah and Jill in this community, is because we've learned a lot about how we can metabolize the chemical that we do use if our soil biology is there to use it up. And like I said, we test outcomes, and the main goal is to be able to grow our crop using soil biology and as little disturbance as possible. So whether that is a herbicide pass or not, the main goal is to make sure that we can provide clean food, clean air, and clean water. And we can accomplish those things and use a herbicide pass if our biology is there to do it. And I know that's not a very popular opinion, but
you got to minimize the disturbance and ultimately mastering the art of doing nothing.
Yeah, we're
is
not
what we
there
like to
yet.
be. We've done it a few times, had a few successful
yes.
do nothing where we've eliminated everything, but not consistently on a year-to-year basis. But there is those occasional times where Mother Nature just clicks and everything falls in place, and we can actually go out there.
Well, and so that you mentioned testing outcomes, and one of the ways to help demonstrate to consumers that they're getting what is important to them is that you can test the pesticide residue on the actual crop. And I know the Axons have done that, and I think you guys are planning to perhaps. And in systems that are very healthy, you do have a zero pesticide residue test on the actual grain because the soil has eaten it up. And because you applied it early, you didn't apply it, shortly before you were going to
right.
harvest it. So those tests become very important, but then also communicating about that. This is a whole new education that has to happen for the consumer, and it's going to take a long time. People are just going to look in their mind for organic because that's what means to them, no chemical for me. That's what they care about. They mostly. So
and everyone does. There's not one
yes.
farmer, not one of us wants to be poisoning the food system. Not one of us.
Right.
And I like that you said that means no chemical for me, which
Right.
is important in our situation. We want to make sure there's no chemical for that consumer. We
And
may
no
use it
for the
somewhere
animal.
on the farm,
Right.
but we want to make sure everything's in place so that doesn't end up in the animal.
We don't want it in the water. We don't want it in the air. Nobody does.
Right.
And the point is, it's not an all or nothing.
Yes.
Okay. And we can do it better. We can use it better. We can use less. And that becomes really important in our system. We would like to be 100% herbicide free. We aren't yet. The solution in there somewhere is in companion planting. But we have not had consistent results with that. It's come a long way.
But we're probably down 80%.
Right.
That's
So
amazing.
eliminated everything. The herbicide's the last thing to go.
That's the only thing we're
And we do have a question from Mark asking about what herbicides do you think are the least harmful and any insecticides?
Oh, we're not using the insecticides.
Right.
Okay.
So we cold turkeyed off of the insecticide and the fungicide, and from our system, from where we were pouring it all on, right? So everything before 2012 was highly, lots of inputs, lots of insecticide, lots of fungicide, lots of trips across the field. The environmental impact of the equipment going across the field that time, or those amount of times is a lot. It's extensive. And it becomes a matter of using less better, right? So do you have it's hard to, now I forgot the question.
Which herbicides are the least harmful?
Oh, that's how I got started. Okay, that's right.
Well, as far as gauge that,
I don't know that answer.
I know glyphosate's talked a lot about
Yeah.
the industry. But it's a phosphate. You know, if we have those phosphate solubilizers there, I believe they can do, you know, they can take that and metabolize that as a phosphate, you know, those phosphate solubilizers, if they're there. But now if we're applying animal manures or high rates of phosphate fertilizer, those populations aren't going to be there to digest that.
We can put them there.
We can put them there and get them going, but that's going to be pulling, you know, it's we've almost got to get to a it's going to be tough to say, but a nutrient deprivation. You know, we've also got to deprive that a little bit of that phosphate to get those communities active and engaged. So when a glyphosate is applied or something, those are hungry, starving, and they're able to.
Which
able to
Jill
chew
has,
that up.
you know, been a great resource for being able to talk about that from the science side, and how the microbial community is able to break down the chemical, metabolize it, and keep it out of the food, out of the air, and out of the water. And so I don't know if there's one that's less harmful than another. I know that our biological communities need to be there and need to be ready for the disturbance that is there. And my job as a farmer for us on our farm is to create as little disturbance as possible so that the microbial community can bounce back from that.
Now,
And so,
when and where that's applied. Now, if that's applied to the soil where those phosphate solubilizers are, that's a good thing. But now if that's applied directly to the top of the wheat 7 days before, 10 days before harvesting, I don't think that's quite such a good thing.
right. I think there's ways, or
There's
there are
ways.
lots of ways that we can be using less and we can using it better. There's a reason that we are desiccant free on the farm. I don't believe we can keep it out of the food.
We're not having that soil contact, that initial with those phosphate,
Yeah.
you know.
I don't think the
initial-
plant can metabolize it the way the biology in the soil can. So that's for us, one of the reasons we are desiccant free.
Because you're pretty limited on what's available to use if we're not doing tillage to the land. I mean, there's not a lot of options out there besides tilling that ground up, and it's going to take that microbial community a longer time period to rebuild those homes and that soil structure and those aggregates and those bacterial glues and all that than it will be to metabolize a phosphate put down in a low concentration.
And like I talked about, we are reliant upon that community to provide us with a strong, healthy plant that is going to produce our crop that year. So as minimal amount of disturbance as we can do to that soil microbiome. And I will tell you that the herbicide, when it comes to weeds, is the lesser of the two evils. So if we're looking at tillage as an option and herbicide as an option for us in a biological system, our system is going to recover faster from a herbicide pass than it will from me going in there and ripping up their entire community.
And then another thing,
population.
we sat in an organics seminar as well, and with the electro weeders and stuff coming about, you know, we're stripper stubble cereal grains around here. We have wind farm in our area, and with those, there's risk of fire.
So yeah, we got a lot of wind and we're dry.
And we're dry, and our whole farm is 3 foot tall wheat straw. So if we get a spark or a fire started, we've got
a serious problem.
serious problem. Then we lose all of our soil armor.
And it's an evolution, right? So we're evolving all the time and finding new ways to do our job better. So and I think that we're getting closer. We're just not there yet.
And I do think too, so you're building all this great work on the farm and now sending it out to other farmers. And I think there's another kind of education that needs to happen toward consumers. And that's what we're trying to do with, we've got our podcast, Tasting Terroir, which I got to have you guys on so that we can talk about this, but from a consumer perspective so that they can understand. And so we'll do that, but also to others of you, you know, I really invite you to take advantage of that. Let me interview you for our podcast. Let me help explain the kind of things that you're doing. It's complex. It's not gonna fit on a package and just, you know, plus people wouldn't believe it if it's there anyway. They're very skeptical about marketing, but we can take a whole hour. We can really discuss, you know, the complexities of this. And that's a big part of what we're trying to do with that podcast is to help people understand some of the science, some of the biology, and understand the outcomes for them. And then how do they find that in the marketplace? What questions to ask for, what things to look for. And then of course we have a webpage on our main public website that links to Guardian Grains, for example, and Craig Cameron's beef, and any of you that want to be on it. You have to be interviewed for the podcast first, but then we'll have a nice little square for you, link, where people can find you. And it's globalfoodfarm.com/betterfood. And so then over time, we'll be able to kind of help people find people that are growing like this, people that are doing the things that they basically want, but they don't know what to look for, or how to find them. So that's a point of this community, trying to help build that communication messaging and drive people that are looking for it, towards you. So sign up for the podcast, Shorty. Even if you're not selling direct to the consumer, I think helping the consumer understand the way that you all farm and how it's different, and what it really is that they should be looking for when they're evaluating the claims of regen, because it's very buzzy, it's everywhere, it means anything and nothing, depending on who's talking about it. So that's all a service that you can be part of, I think, in building a consumer base that will want what you're doing, that will value what you're doing. So, all right. Well, this is wonderful. Great information. Thank you so much for sharing it, DeAnna and Kelly, and great job. Great presentation.
Thanks for letting us come on and talk about it.
Oh my goodness. No, it's a wonderful part of our community. And as I mentioned, this presentation will be in our RegenFlix library, and there are many other great ones there too, to dive in and learn from. So thank you all so much. Really appreciate it.
Thanks very much, Sarah. Have a great day. Thanks, everyone.
Thank you.
Bye.